Hello, my dear Karnataka Pharma champions. Welcome back to KPC classes. Yes, today in this video we are going to discussion about mechanism of action of penicillin antibiotic. Yes, okay, yeah. So before going to mechanism of action, first we will see some introduction, some basic introduction about penicillin antibiotic. Yes, penicillin already most of you are well known about this drug. Penicillin, it is an antibiotic. Okay, this is the first discovered antibiotic. It is used clinically. Okay, so this is the first discovered antibiotic. Penicillin, it is discovered by penicillin. It is discovered by Alexander Fleming. Alexander Fleming. Okay, in the year 1928. In the year 1928, the Alexander Fleming, he discovered penicillin antibiotic. Okay, so he discovered this antibiotic from the fungi Penicillium notatum. Penicillium notatum. Penicillium notatum. Okay, but now currently penicillin antibiotics, they are obtained commercially from the Penicillium chrysogenum. Penicillium chrysogenum. Okay, yes. So before starting, Alexander Fleming, he discovered penicillin antibiotic from the fungi Penicillium notatum. But commercially now they are obtaining from penicillin antibiotic, they are obtaining from Penicillium chrysogenum. Okay, yes. So this penicillin antibiotic, they consist of two rings. They are made up of two rings. Okay, so first is beta lactam ring. Penicillin antibiotic, this is comes under beta lactam antibiotics. Means it consists of beta lactam ring and thiazolidine ring. Thiazolidine ring. Okay, and these two rings are attached with one amide linkage. Amide linkage. This is the what? Chemical structure of penicillin antibiotic. Okay, so due to the presence of this beta lactam ring, it will shows effective action against bacteria. Okay, so this is the main ring, beta lactam ring. It is the needed for, essential for showing action against bacteria. Okay, so now we will see the exact mechanism of action of penicillin antibiotics. Okay, yes, penicillin antibiotics, they act by inhibiting cell wall synthesis in the bacteria. They act by inhibiting cell wall synthesis. Okay, how they will act? By inhibiting cell wall synthesis. Okay, ಬಟ್ ಸುಮ್ಮನೆ ಏನ್ ಮಾಡ್ಕೊಡ್ತೀರಾ ನೀವೆಲ್ಲ ರಟ್ಟ ಹೊಡೆದ್ಬಿಡ್ತೀರಾ, ಕಂಠಪಾಠ ಮಾಡ್ತೀರಾ. ಬಟ್ try to understand the concept. Yes, penicillin antibiotic, they inhibit cell wall synthesis, so that's why bacteria will going to die. This is the what mechanism of action of penicillin antibiotic. But first you should know how the cell wall synthesis takes place in bacteria. Then only you will get idea how the exact penicillin antibiotic, they inhibit cell wall synthesis in the bacteria. Okay, so before going to mechanism of action, first we will see cell wall synthesis in the bacteria. Cell wall synthesis in bacteria. Okay, cell wall synthesis in the bacteria. Yes, gram positive bacteria and gram negative bacteria, both are composed of cell wall. Comparatively, gram positive bacteria, they have a thick cell wall that is made up of peptidoglycan layer. Peptidoglycan. Okay, so due to the presence of this peptidoglycan, cell wall becomes more strong. If cell wall is more strong, then automatically bacteria will also get more strong. If there is no cell wall, then bacterial cell is weak. Okay, so this is the concept actually. Okay, yes. Now we will see how the exactly cell wall synthesis takes place in the bacteria. Okay, as you can see, bacteria first they synthesize N-acetyl glucosamine. N-acetyl glucosamine, or it is you called UDP N-acetyl glucosamine. UDP means uridine diphosphate. Uridine diphosphate. It gives energy and the movement for the NAG molecule. NAG means N-acetyl glucosamine. As you can take N-acetyl glucosamine. Means this is what? Completely sugar. It is completely made up of sugar portion. Okay. UDP N-acetyl glucosamine. First, bacteria, they synthesize UDP N-acetyl glucosamine. Here, one enzyme which is present inside the bacteria, that is phosphoenol pyruvate transferase. Phosphoenol pyruvate transferase. This enzyme, it is present inside the bacteria. It will be helpful for the conversion of UDP N-acetyl glucosamine to UDP N-acetyl muramic acid. NAM means muramic acid. N-acetyl muramic acid. Muramic acid. First, bacteria, they synthesize UDP N-acetyl glucosamine and it will be get converted into UDP N-acetyl muramic acid with the help of enzyme phosphoenol pyruvate transferase. Okay, and this N-acetyl muramic acid, this is also made up of sugar portion. Along with that, it also contains a lactyl group. Okay, so due to that, here one pentapeptide chain will be going to get attached here. Pentapeptide chain. Pentapeptide means what? First, we will discuss pentapeptide chain. Pentapeptide chain. This is also synthesized by bacteria. Pentapeptide means, penta means 5. Peptide means this is made up of amino acid. Means here the pentapeptide chain, it is made up of 5 amino acid. Those 5 amino acids are L-alanine, D-glutamate, and L-lysine, D-alanine, and D-alanine. Okay, so this is the what? The composition of pentapeptide chain. Okay, so now we will discuss how this pentapeptide chain will be formed. First, bacteria, they synthesize. See, we will discuss, okay, one by one. First, bacteria, they synthesize one amino acid, that is in the L form, L-alanine. Bacteria, they synthesize one amino acid, that is L-alanine. So this L-alanine amino acid, levorotatory, L means what? Levorotatory. This amino acid, L-alanine amino acid, this is less stable and it is comparatively, it is less strong and it is easily recognized by some enzymes which are present inside the human bodies. It is easily recognized by some enzymes and it will be easily destroyed. So that's why bacteria, they have to make it strong. So that's why they will convert L-alanine into D-alanine. L-alanine into D-alanine. For this conversion, there is a requirement of one enzyme, that is alanine racemase. Alanine racemase. This is the what? The needable enzyme for the conversion of L-alanine into D-alanine. Don't worry. Okay, so one by one I heta hogtini correct agi. First, bacteria, they synthesize UDP N-acetyl glucosamine. This is completely sugar portion, and phosphoenol pyruvate transferase, it will convert UDP N-acetyl glucosamine into UDP N-acetyl muramic acid. Okay, so to this UDP N-acetyl muramic acid, there is an attachment of pentapeptide chain. So this pentapeptide chain, it is combined of 5 amino acids. So this pentapeptide chain, it is formed by first bacteria, they synthesize L-alanine amino acid, but this is a less strong, less stable, and due to easy destruction, bacteria, it will be get converted into D-alanine. For this conversion, there is a requirement of one enzyme, that is alanine racemase. Okay, after that, two D-alanine molecule will be attached with the help of enzyme alanine ligase. With this type of enzyme, two D-alanine molecule going to combine. Okay, so this is the what? The exact composition and synthesis of pentapeptide chain. Is it clear, student? Yes, pentapeptide chain, it is made up of 5 amino acid. How it will be formed? First, bacteria, they synthesize L-alanine amino acid, but due to the less stable bacteria, it will be get converted L-alanine amino acid into D-alanine. For this conversion, there is a need of an enzyme, that is alanine racemase. After that, two D-alanine molecule, they will get combined with the help of enzyme alanine ligase. Okay, so this is what the exact composition of pentapeptide chain. So this pentapeptide chain will come and attach to the UDP N-acetyl muramic acid. Okay, so after that, so this is what this all exact process, they are occurring inside the cytoplasm, inside the cell. Okay, inside the bacterial cell. Okay, inside means cytoplasm alli ಆಗ್ತಾ ಇತ್ತು process correct ಆಗಿ. But now here this is a cell membrane. Consider this is a cell membrane. Okay, this is a cell membrane. Now on cell membrane there is a one lipid carrier membrane. Lipid carrier membrane. Lipid carrier membrane. This is known as bactoprenol. Lipid carrier membrane. This is known as what? Bactoprenol. Okay. So this bactoprenol, this lipid carrier membrane, it will be helpful for the transfer of this UDP N-acetyl muramic acid pentapeptide chain to the outside of the cell. It will be transport ಮಾಡುತ್ತೆ. Because this is UDP N-acetyl muramic acid pentapeptide chain. This is a completely hydrophilic. For crossing cell membrane completely is lipophilic. Okay, cell membrane ಏನಿರುತ್ತೆ lipophilic ಇರುತ್ತೆ. For crossing lipophilic membrane, the molecule should be lipophilic only. But here UDP N-acetyl muramic acid pentapeptide chain, this is a completely hydrophilic. For transporting this membrane, here it will be help. Bactoprenol will be helps. Okay, so bactoprenol, this is a lipid carrier membrane, like boat ತರ ವರ್ಕ್ ಮಾಡುತ್ತೆ. ಓಕೆ, ಸೊ ಒಂದು ಸ್ವಿಮ್ಮಿಂಗ್ ಮಾಡಬೇಕಾದ್ರೆ ನಮ್ಗೆ ಸ್ವಿಮ್ಮಿಂಗ್ ಬರೋದಿಲ್ಲ. ಓಕೆ, ಸೊ ಅವಾಗ ನೀವು ನದಿಯಲ್ಲಿ ಯಾವ ತರ ಕ್ರಾಸ್ ಆಗ್ತೀರಿ? ವಿತ್ ಯೂಸಿಂಗ್ ಬೋಟ್. ಓಕೆ, ಸೊ ಅದೇ ರೀತಿ ಇಲ್ಲೊಂದು ಬೋಟ್ ಇರುತ್ತೆ, ಅದು ಯಾವ್ದು ಅಂದ್ರೆ ಬಕ್ಟೋಪ್ರಿನಾಲ್. ಈ ಬಕ್ಟೋಪ್ರಿನಾಲ್ ಏನ್ ಮಾಡುತ್ತೆ? UDP N-acetyl muramic acid pentapeptide chain ಏನ್ ಮಾಡುತ್ತೆ? ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಕ್ರಾಸ್ ಮಾಡಿಸುತ್ತೆ. outside of the cell wall, outside of the cell membrane cross ಮಾಡಿಸುತ್ತೆ. So it will, this bactoprenol, it will remove UDP molecule. It will remove UDP molecule. It will take only NAM pentapeptide chain. It will take only NAM pentapeptide chain and NAG molecule, N-acetyl glucosamine. See, concentrate. Bactoprenol, it will remove UDP molecules because bactoprenol ಏನ್ ಮಾಡುತ್ತೆ? UDP molecule remove ಮಾಡಿಬಿಡುತ್ತೆ. He wants only bactoprenol. He pick up only NAM pentapeptide chain and NAG molecule. Okay, that will combine NAM and NAG. They will be get combined and they will be transferred outside of the cell membrane. Okay, NAM and NAG. Sorry. Okay, NAG and NAM pentapeptide. Okay, pentapeptide chain. We will consider NAG as G and NAM as M. Okay, short form ನಲ್ಲಿ ಬರುತ್ತಾ ಹೋಗೋಣ, ಅಂದ್ರೆ ಕ್ಲಿಯರ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. Okay, so once NAG and NAM pentapeptide chain, they will combine, and after that outside of this cell membrane they will elongated. Okay, so this chain form, it will be elongated, and NAG, NAM. Okay, NAG, NAM. I am writing here P. P means what? Pentapeptide chain. Same like this chain elongation takes place. For this elongation of chain, there is a one enzyme that will be helpful, that is transglycolase. Transglycolase. Okay, so this transglycolase enzyme, it will be helpful for the elongation of pentapeptide chain, means peptidoglycan chain. This is known as peptidoglycan chain. Okay, here the elongation of chain takes place with the help of enzyme transglycolase. Okay, after that this chain, it will be going to cross linking ಆಗ್ತಾ ಹೋಗುತ್ತೆ, like uramic acid pentapeptide, NAG, NAM pentapeptide. Okay, pentapeptide. So like that, pentapeptide. So here there is a combination. Okay, like this cross linking will be takes place between two pentapeptide chain molecules. There is a removal of last D-alanine molecule. Okay, so completely there is a removal of last D-alanine molecule remove ಆಗಿಬಿಟ್ಟು ಏನಾಗುತ್ತೆ? There is a bond formation takes place between two pentapeptide chain molecules. Okay, so due to that there is a cross linking of peptidoglycan chain. ಓಕೆ ಸೊ ಈ ಕ್ರಾಸ್ ಲಿಂಕಿಂಗ್ ಜಾಸ್ತಿ ಇತ್ತು ಅಂದ್ರೆ ಫಾರ್ ದಿಸ್ ಕ್ರಾಸ್ ಲಿಂಕಿಂಗ್ ದೇರ್ ಇಸ್ ಎ ರಿಕ್ವೈರ್ಮೆಂಟ್ ಆಫ್ ಅನ್ ಎಂಜೈಮ್ ದಟ್ ಈಸ್ ನೋನ್ ಆಸ್ ಟ್ರಾನ್ಸ್ಪೆಪ್ಟೈಡೇಸ್. ಓಕೆ ದಿಸ್ ಇಸ್ ಆಲ್ಸೋ ನೋನ್ ಆಸ್ ಪೆನ್ಸಿಲಿನ್ ಬೈಂಡಿಂಗ್ ಪ್ರೋಟೀನ್ ಅಂತಾನು ಕರಿತೀವಿ ನಾವು. ಸೊ ದಿಸ್ ಟ್ರಾನ್ಸ್ಪೆಪ್ಟೈಡೇಸ್ ಇಟ್ ವಿಲ್ ಬಿ ಹೆಲ್ಪ್ಫುಲ್ ಫಾರ್ ಕ್ರಾಸ್ ಲಿಂಕಿಂಗ್ ಆಫ್ peptidoglycan chain. Okay, if cross linking is high, then cell wall becomes more strong. If there is no cross linking, then cell wall becomes weak. Okay, here now penicillin antibiotics they acts on transpeptidase enzyme and that will be inhibited. So if there is no transpeptidase, then there is no cross linking of cell wall peptidoglycan chain. If there is no transpeptidase, then there is no cross linking of peptidoglycan chain. If there is no cross linking of peptidoglycan chain, then cell wall becomes weak. If cell wall becomes weak, then bacteria will go into die. This is the exact mechanism of action of penicillin antibiotics. Okay, is it clear, student? Yes, I will explain once more. Okay, now you can clearly understood. Okay, see, yes, as you can see here. Yes, first what I said, bacteria they synthesize UDP N-acetyl glucosamine, that is G means what? N-acetyl glucosamine, and it will get converted into UDP N-acetyl muramic acid with the help of enzyme phosphoenol pyruvate. Okay, and after that pentapeptide chain it will be attached to the UDP N-acetyl muramic acid. Okay, this pentapeptide chain it is made up of 5 amino acid. Okay, first L-alanine it is formed. After that it will get converted into D-alanine. Okay, this is the what? The combination of 5 amino acid of pentapeptide chain. It will be attached to the NAM muramic acid. Okay. After that, here are the bacterial, as you can see here in this picture. This is a lipid carrier membrane. So these formed UDP N-acetyl muramic acid, these are hydrophilic. So that's why it will be helpful for the transferring through the cell membrane. Okay. So this bacterial, it will pick UDP N-acetyl muramic acid. It will be remove UDP molecule. UDP molecule will be get removed from the N-acetyl muramic acid. It will take only N-acetyl muramic acid pentapeptide chain and N-acetyl glucosamine. Okay, as you can see here in this picture, N-acetyl glucosamine and N-acetyl muramic acid pentapeptide chain, and that will be transferred outside the cell. Okay, this is N-acetyl glucosamine, N-acetyl muramic acid pentapeptide chain. Okay, after that the transglycanase enzyme it will be helpful for the chain elongation. As you can see here, this is the chain elongation. N-acetyl glucosamine, N-acetyl muramic acid, N-acetyl glucosamine, N-acetyl muramic acid. This is what? Chain elongation. Okay. After that, cross linking will be takes place with the help of enzyme transpeptidase. As we can see here, this is the cross linking, like N-acetyl glucosamine, N-acetyl muramic acid, like this cross linking. Pentapeptide chain cross linking will be takes place. Okay. Now this here the penicillin antibiotic they inhibit transpeptidase enzyme. Penicillin antibiotic they inhibit transpeptidase enzyme. If there is no transpeptidase, then there is no cross linking of peptidoglycan chain. If there is no cross linking of peptidoglycan chain, then there is no cell wall strong. Then cell wall becomes weak. Then bacterial cell will go into die. This is the exact mechanism of action of penicillin antibiotics. Okay, ಇದು ಕ್ಲಿಯರ್ ಆಯ್ತಾ ಸ್ಟೂಡೆಂಟ್? ಎಸ್ ಎಸ್, ಇದೇನಾದ್ರು ಕ್ಲಿಯರ್ ಆಗಿತ್ತು ಅಂದ್ರೆ ಕಮೆಂಟ್ ಬಾಕ್ಸ್ ನಲ್ಲಿ ಎಸ್ ಕ್ಲಿಯರ್ ಅಂತ ಬರೀರಿ. ಓಕೆ, ಸೊ ಎಸ್ ಇನ್ನು ಯಾವ ರೀತಿ ನಿಮ್ಗೆ ಟಾಪಿಕ್ಸ್ ಕ್ಲಿಯರ್ ಆಗಿ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮಾಡಿಸ್ಬೇಕು, ಪ್ರತಿಯೊಂದು ಟಾಪಿಕ್ ನ ಸರ್ಟನ್ ಟಾಪಿಕ್ಸ್ ನ ನಿಮ್ಗೆ ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ ಪ್ರತಿಯೊಂದು ವಿಡಿಯೋ ಅಪ್ಲೋಡ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಓಕೆ, ಎಸ್, ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್ ಸ್ಟೂಡೆಂಟ್ಸ್.